Chapter 6 in our textbook, there are different terms and descriptions of growth conditions or growth requirements for different kinds of microorganisms. In particular, we're usually thinking of bacteria as our example microorganism. Most prokaryotic bacteria, or prokaryotic cells, like archaea, will grow and divide by binary fission. Even the cyanobacteria will grow and divide by binary fission. We have seen some exceptions. Myxobacteria, Hyphomicrobium, Caulobacter all grow by budding. But most prokaryotic cells undergo binary fission. One cell gives rise to two. It's a very simple process. No cytoskeletons, no anaphase and telophase, simply one cell that begins to elongate. A copy of the genetic material is manufactured or produced and sort of spread or deposited to one end of this elongated cell. The cell wall and plasma membrane invaginate or pinch off the two ends of this newly forming cell so that a cross-wall forms and completely separates the two halves. Eventually, those two halves break away, and you now have hopefully two identical daughter cells compared to the original cell that we started with. Now again, if we take a look at both cocci and bacilli as they're growing and reproducing, for the cocci, because they have a nice round spherical shape, we can have planes of cell division that are 1, 2, 3, or more than 3. And the planes of cell division for rod-shaped bacilli, or bacillus cells, is essentially in one plane, going from 1 to 2 and 2 to many more in just one direction. Typically, when we're counting the number of cells produced in binary fission, one cell makes 2, 2 cells make 4, 4 make 8. That's described as exponential or logarithmic growth. If we look at those numbers, we can find that the numbers of cells produced can be expressed as a power of 2. So, and we call these generations. The first generation to the second would be 2 to the first power. The second generation to the third would be 2 to the second power, and on and on. So if we take a look at the visual representation of numbers, we can see that fairly quickly we can start with one bacterium and wind up with thousands of bacteria produced from that original first cell. Exponential growth or logarithmic growth can also be depicted graphically by a line graph. And we can divide the growth up into 4 stages or phases. The first stage is the lag phase. If we look inside these cells during lag phase, we're going to find when we graph out those numbers over a period of time, counting the number of bacterial cells, in the lag phase there's no increase in numbers of cells. But inside these cells, they are metabolically very active. They're producing all the building blocks and all the materials needed to go into the second phase, which is that exponential or logarithmic phase of growth. When we start to look at the cell numbers during exponential growth, there's a tremendous increase in the number of cells produced. Because now we're going through that 1 cell makes 2, 2 cells make 4, 4 make 8 period of growth. Eventually, some of the food is going to start to run out. When food is becoming limiting, and we don't have enough food to start making more cell parts, then we're going to see that there will be a plateau. We won't see any additional cells. In fact, the number of dead and dying cells will be pretty much equivalent to the number of newly synthesized or newly made cells. So that's the stationary phase. Now we're getting to a point where we don't see any change in cell numbers. If you don't transfer your bacteria to some new food, and they all need fresh food, just like we do, eventually you're going to find that the cells are more numerous that are dead and dying than the ones that are newly being produced. This is the death or decline phase. Now, if we would actually look at the cloudiness of a broth culture, or simply look to see if a colony is still sitting on the surface of the agar, we might find that everything's dead, even though we can still visibly see the remnants of the cells that were there. So just because a cell is dead doesn't mean it necessarily is going to disappear in our culture, especially on our agar plates. If the culture is too old, even though you can still see the colony and it still looks fresh, if you try to take a little sample of it and put it into some fresh food that we call subculturing, there's nothing there alive or vulnerable or viable to go on and pass on to the next generation. So it is important to maintain cultures of bacteria and constantly give them fresh food. When we're describing oxygen requirements, we might use the phrase obligate aerobe. Obligate means must have. Aerobe, we're talking about oxygen. So an obligate aerobe must have oxygen for growth. These organisms use what we call cellular or aerobic respiration and require oxygen as a terminal electron acceptor. In Unit 2, we go through metabolism in more detail, so it will make more sense exactly what those phrases mean. But cellular or aerobic respiration is essentially what's occurring in your cells. So we can use these kinds of bacteria to study those types of metabolic processes. If you take a look at the test tubes, the first test tube in this photo on the left is actually a control tube. And the pink color near the top of this liquid, which is a broth medium, actually has an indicator that tells us where is the oxygen in this liquid. And an obligate aerobe, because it has to have oxygen and lots of it, grows right on the top of the broth liquid. If we have oxygen requirements and describe an organism as a facultative anaerobe, it's an almost misleading phrase because anaerobe implies without oxygen or no oxygen. When you add a prefix a or an to a word, it essentially negates or says no to whatever the root word is. So no oxygen, that's what's implied by the word anaerobe. But facultative, well, that simply means these organisms can easily shift gears. If oxygen is present, these cells will grow in a thick, dense culture and use the oxygen if it's present. If oxygen is absent or missing, these organisms will still grow. They'll just use alternative metabolic processes that don't require oxygen. In fact, if we look at the test tube, that's essentially the second one from the right, and compare the facultative anaerobe growth, which is very, very cloudy in the bottom half of the tube, actually the bottom 90% of the tube, versus the growth in the tube that has the obligate aerobe. We can see that the obligate aerobe cannot even grow below the surface of the broth. The facultative anaerobe, even though it's much lighter growth than the top, we still have growth in from the top to the bottom the test tube. It's just that the growth at the very top of the broth, where oxygen is concentrated and available for the cells, we have much thicker luxuriant growth in the presence of oxygen versus the growth of the organism where oxygen is absent. The reason for the difference in growth? If oxygen is available and we're using aerobic cellular respiration, we actually generate more energy-yielding molecules in the form of ATP, 38 per molecule of glucose, versus if oxygen is not available and the cells that are in this broth medium are simply growing by fermentation, which yields 2 molecules of ATP per molecule of glucose. Now, in Unit 2, we go through more detail about metabolism. But needless to say, facultative anaerobes have the advantage. Whether oxygen is absent or not, they keep growing as long as they have a source of food. Another term that's used to describe organisms that don't use oxygen at all, and in fact are not even bothered or don't even know that oxygen is absent or present, are the aerotolerant anaerobes. Aerotolerant anaerobes are growing by fermentation, or do not even sense oxygen is present or absent. And if you take a look at the growth pattern between the fourth tube on the right and the second tube on the right, we'll see that the difference in growth patterns for the facultative anaerobe versus the aerotolerant anaerobe, if we look at the top of the test tube of broth, the facultative anaerobe has very thick, luxuriant growth. Because it's growing by cellular respiration, we get more cells, more energy produced, versus the aerotolerant anaerobe, which is only growing by fermentation. So we have the same sort of pattern of growth from the top to the bottom of the test tube of that kind of cell, versus the very difference between the top and the bottom of the facultative anaerobe. Another phrase that we can use to describe oxygen requirements of certain kinds of prokaryotes is obligate or strict anaerobe. An obligate, remember, means must have. Anaerobe, no oxygen. For these types of organisms we call obligate or strict anaerobes, if oxygen is present, these organisms are missing or do not produce the kinds of key enzymes that are needed to tolerate oxygen. So the oxygen actually kills these organisms. And they still grow by fermentation. But what you'll find is if you take a look at the test tube that's the second one from the right, so the tube that's on the far right is actually got chunks of pieces of something in the test tube, and then the second tube from the right has growth just below where we would find oxygen diffusing into the liquid. So you should see at the top of the liquid in the test tube second from the right, there's essentially nothing growing in the top of that liquid medium because this organism cannot tolerate the presence of oxygen. This slide is a summary of all of the terms that we've used to describe oxygen requirements. Obligate aerobes, facultative anaerobes, obligate anaerobes, and aerotolerant anaerobes. The description in the pictures will give you additional illustrations to hopefully remember the differences between these different categories. Plus, we have one more category called the microaerophiles. We've actually already described microaerophilic growth in one of the introductory Chapter 1 slides. Micro meaning tiny, aerobe meaning oxygen. These organisms only need a small amount of oxygen to grow, and typically will grow just underneath the surface of the broth container or the broth medium in which they're being grown. So, please be sure that you understand and can recognize the different definitions and different oxygen requirements for all 5 categories of oxygen requirements.